So after the talk of Mauro, we're going to have a coffee break. And after that, we're going to go to the first floor. There is a space where there are tables there, and try to cluster through some kind of affinity and build groups of 4 people. And in this hour and a half that we're going to have downstairs, we're going to be hanging out with you guys, answering questions, but specifically trying to help you choose some paper you would like to present. So we put some titles that could be more or less representative, but you can download them, look at the abstracts, and talk to us about that. The idea is that then you will leave, and maybe later after dinner with your friends, you'll end up choosing some paper. The idea is that by tomorrow you tell us which paper you decided to do. We'll micromanage if there are many people trying to work with the same paper, but I don't think that's going to happen. But this is the dynamics. And there, again, the format is completely free. If you want to reproduce some simulation because you brought your laptop and you feel comfortable with that, and you're in a PhD program, you think it's a piece of cake or something, it's okay. If you just want to discuss it, if you want to make a nice presentation, the format is pretty, it's pretty free. But I guess that, a small PowerPoint presentation with some figures, even if it's just by taking them out of the paper, that would be the most safe thing. You're going to have the input also of Bill Bialek and Maxi San Miguel, which is an interesting opportunity. But in any case, be careful. The only thing that we ask you to do is to be very careful with the timing, because we have many groups, and the idea is that we're going to have only 15 minutes, so...
Almost critical.
So the idea is that basically try not to go over the time of the 15 minutes, because we won't have enough time otherwise. So thank you very much.
It's going to be fun. It's going to be very, very nice. Okay. So far so good. First part was called The Rise of Direct Percolation, and I gave you all the good news, everything worked beautifully. By the time I had published that paper on the dynamic range, I loved my talks because they always ended like this, with the positive derivatives, like, yeah, everything is good, it explains the data, nice. But then, of course, life goes on. And if you're interested in the brain enough, you'll very quickly realize that it's a very, very, very complicated stuff. And therefore, there's no way the story could be so simple as I'm telling you. So please bear with me. The second lecture hurts a little bit more than the first one, okay? I'm just warning you in advance. It hurts because it's not intentionally confusing, but unavoidably confusing, because we were confused ourselves. More or less the whole field was, with some contradictory results, some things that we don't understand, and some theories that start not to work. And if you, by the end of the second lecture, if you are puzzled, then I'm satisfied, because so were we. We were a bit puzzled. There's lots of experimental results, because it's very easy to make theories about the brain, but the brain has to agree. And essentially, it didn't in this second lecture, okay? So please bear with me. I know it's going to hurt, but it's for a good reason. Discomfort can be good, because then it moves you forward. So we had published that paper on the maximum dynamic range on a model. And 3 years later, we had this confirmation in Dietmar Plann's lab, but again, it's in a dish. The whole point, the whole reasoning behind the critical brain hypothesis is that it should be useful for behaving animals, right? So I had published that paper, and then Siddhartha Ribeiro, my good friend in Natal, a neuroscientist, a biologist, came to me and said, okay, here's some data from real animals. Show me the criticality there. in my animals, in my lab. He challenged me. I was going to say I am a theoretical physicist. I'm not sure I am still, but I was a theoretical physicist. So eventually you get a DVD full of data from live rats. Well, what do you do with that? So, Tiago Ribeiro was also a physicist, also a theoretical physicist by then, took up the challenge of analyzing the data. Today he's doing experiments at the NIH with Dietmar plants, so he became a neuroscientist eventually. And so in collaboration with many people, Dante Calvo, also Fabio Cacheta, Miguel Belchior, we came to this thing. So this is what we're talking about here now, instead of those simple models and petri dishes. So Siddhartha had this experiment from a different paper in 2007. It had nothing to do with criticality, nothing at all. Siddhartha is interested in sleep. So the main difference between what I showed you before as far as experimental data goes and what I'm showing you here is that here we talk about freely behaving rats, which stay in a box like this. They can do whatever they want, whenever they want. So they can go across the main sleep-wake stages of the sleep-wake cycle. So WK is waking, like you're supposed to be now. And slow-wave sleep, that's the kind of deep sleep where you do not dream. And REM sleep, REM stands for rapid eye movement, when we humans are dreaming, and presumably so are other mammals like the rats. So they go around the cycle however they want. And for different reasons, Siddhartha was also implanting them in different regions of the brain. In the primary visual cortex, because those experiments were done in the dark, therefore you should see nothing extraordinary going on in the visual cortex. In the primary somatosensory cortex is tied to the rat's whiskers, which is the most important part of its tactile sensations, like the paws. And hippocampus, which is a brain region related to memory, information, and localization. So the experiment is like this. The rats are in a cage to which they are well adapted, they are used. So this is not a novel environment. It's they know this thing. And they are implanted with electrodes in all those regions, okay? And then Siddhartha introduced 4 novel objects to the rats. The rats go crazy because they get very curious about it. They explore the object, they touch the objects, they try to explore the objects for 20 minutes. Everything is video recorded with infrared cameras. And then you remove the objects and they stay there for another 3 hours. And the experiment reports in these papers are very, very extensive. So several different animals are tested. They are then sacrificed at different stages of the experiments to measure gene expressions, to see where memory about these objects are formed, where, how long it takes, et cetera, et cetera. So it's an experiment designed for something else, not my purposes. He just had it, this kind of thing for different animals. So we have 3 phases of the experiment. You have pre-exposure, during exposure to novel objects, and post-exposure to novel objects. So please, keep in mind the mess that this is, because essentially now I have 3 behavioral states, 3 brain regions, and 3 phases of experiments, which means that whenever Tiago Ribeiro would come to my door to show me plots of anything, or whatever you think of, it was 27 versions of those plots, 3 times 3 times 3 possibilities, right? I'll show you soon one version of these 27 plots. And here they are measuring spikes, not local field potentials. So in order to measure spike, you have an electrode here in the brain. Brain, have an electrode, have another electrode here. So depending on how you filter your signal, you can detect these spikes, which again last about 1 millisecond. So you have, in order to see the signal, you have to bend past just letting the high frequencies go through. And then you have the signal here. Say in these electrodes, which goes like this. And here, it goes like this. So you can see that sometimes you have coincident spikes here. You can make some types of triangulation and try to detect, okay, this spike which appears in this electrode and this electrode came from the same guy, and I can tell you this is one specific neuron. This guy has a different shape than this one, so this comes from a different neuron. So we can do there's a whole set of techniques in order to be able to tell whether what you detect is really a spike or not. Okay? There's a whole art behind this, which I'm not going through, but you can do it. But essentially what I have is just binary events for each neuron from tens of electrodes with different numbers depending on the rat. Sometimes you implant the electrodes on the rat and it doesn't work. It doesn't work for all the electrodes. Thais, who's there, she's an expert on that. She can tell you how hard it is. So you're measuring spikes. It's a different kind of signal. And also, we were interested about anesthesia. So rats were also anesthetized with this particular kind of anesthetic, ketamine, xylazine. I know that physicists, typically, you're probably thinking, I couldn't care less what kind of anesthetics. For me, it's just anesthesia, right? Yeah, I was forced to learn this lesson the hard way. So there's no such a thing as anesthesia in biology. Which anesthesia? Ketamine, xylazine, propofol, urethane, you name it. They're different. Okay? Just keep this in your mind. They're different. Here I'm talking about deep doses, heavy dose of ketamine, xylazine. It's a kind of anesthetic that you can wake up from and still be alive. So do the animals. Okay? What?
It's a dissociative anesthesia too. It's a dissociative anesthesia, so you are kept away from your sensory information.
Yes, you're kept away from your sensory information. It was used in World War II, right? People would have hallucinations and this kind of anesthetic. So this is what it looks like, if you want to look at it. So this is a typical plot that you see in neuroscience. This is called a raster plot. In the horizontal axis, you have time. In the vertical axis, you have, for each line is a different neuron, and each mark here is one spike. So I reduced this thing to what a physicist doing stochastic process would call a point process, for instance, something like that, right? So I have events. And this is what an event in waking look like. Some activity like this. No real science. Slow wave sleep has such a name because it looks like slow waves. You have some rhythm here. REM sleep looks a little bit like waking. And in fact, it's something it's considered by some people like one kind of consciousness. If you're awake, if you are dreaming, you are conscious of your dreams. This is what ketamine xylazine anesthesia looks like if in a heavy dose. And this is one example of V1, and this is S1. This is some time scale, 250 milliseconds here. Okay, so I got this data from Siddhartha as a gift. That's how collaboration starts. What do? You heard what I told you so far. You would do the same thing that I did. Well, people in criticality, what do they look at? Power laws. They look for power laws in size distributions, right? That's what they do. That's what I've shown you all the time. Power laws, power laws, power laws. Yeah, let's look for power law. So how I went to the power laws here? First of all, separation of time scales is not very clear here. It is clear here in anesthesia. There's some clear silences here, I think, right? We can all agree that there are some silences here. But here, maybe here, but here I don't see any silence at all. So this is a problem to start with from a conceptual point of view. But what we do is operationally, we define an avalanche. Okay. We do the same thing as I was telling them here before. I have a time series with some, hold on, this is bad. Let me start again. I have all the spikes. And now I want to calculate avalanches. Well, like I said before, if I choose a beam which is this size, it's a very bad beam, because I have infinite avalanches of size 1. Do you agree? If I choose a beam this size, I have one single avalanche of size infinity. It's also bad. How do I know it's bad? Because I look and see it's bad. And how do I see it's bad? Because I'm seeing that this is very large and this is very small. As compared to what? As compared to the interval that I'm seeing here. So what they do operationally, what we did, everybody does, is take these intervals here, take an average of them, and use the average size, average interval to define your beam. And then once you have beams, you can bin your data and eventually find, I find one silence here, silence, da, 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 da. Okay? This is what is being shown here. And once you have a beam, yes?
The frame make difference?
What?
The frame you use to beams, like start,
Where we start, you mean?
yes.
It doesn't matter much. No. Because the maximum you can vary is one beam size, right? It doesn't matter much. So, are we okay with what we did? Just operationally define an avalanche, even if it's not clear how to deal with lack of separation of time scales. And we calculate a different time beam for each situation. So V1, waking, is one time beam. V1, slow wave sleep, is a different time beam. So we're just trying to be fair to the data. Try not to impose any artifact to situations which could be different. So to each different situation, we recalculate the beam and we do the avalanche analysis. So what is the first thing that we should do? Let's look at, this is, after you do this, this is a time series of avalanche sizes, S, as a function of awake time for the awake state on one of the largest awake windows of one rat. You see that this looks promising in the sense that this vertical axis is in a log scale, and I'm spanning at least 2 orders of magnitude, which is good, because if I want a power law distribution of something, it should spread along more than 1 order of magnitude, it should have a fat tail. So just by looking here, one gets hopeful. So I could get a power law. That's what it looks like. So let's do the distribution for all the 27 combinations times 8 rats and see the beautiful power laws. Then we get famous and rich. That's all, right? So you do the probability distribution of those power law, of those avalanche sizes for the 27 combinations. Again, 3 brain regions, 3 behavioral states, waking, slow wave sleep, and REM sleep, and 3 stages of experiment, pre-exposure, during exposure, post-exposure. 27 possibilities to obtain power law. You make the plot, and you get these beautiful power laws. This is a log scale, this is a log scale, and these are perfect straight lines. As people in the 90s would do, people in the 90s were interested in self-organized criticality. They would take whatever appear in their tables, and make plots like this, just with the symbols, and then cross, just pass a straight line, and argue that it's a power law. Obviously, these are not power laws. And please note that there are 27 plots, okay? This is 3 by 3, 9, times 3, in 3 colors, and that's 27, okay? So this is V1, S1 hippocampus, pre-exposure, during exposure, post-exposure. These are not power laws. This is the first obvious fact that we had to humbly admit, swallow. But there's something interesting about them. Despite the very different behaviors of REM and slow wave sleep and waking, once you choose a different beam, you get this distribution which look very much alike. This is something that we noticed. It's very interesting, and it's not an artifact of the method, because if you shuffle the data, now you take your spikes and you shuffle, just keeping the same rate that they have, but destroying correlations, everything disappears, and these fat tails, which are not power laws, but are still fat, they disappear. So you were puzzled. We didn't know what to do. We didn't know what to do, and we came up with an idea. Let's say I'm very faithful to criticality, which we are. I don't know. And I think the system is critical. Why then don't I see power loss? It could be the following, and I'm going to argue with that. It could be the following. Assume this is your brain. It's a piece of your cortex. It's nearly two-dimensional anyway. So I put an electrode here, and then another one here, another one here. If I measure local field potentials, like Dietmar Pence was measuring in the original paper that I talked to you about, you're measuring electroactivity all this way. If you're measuring local field potential here, you're also measuring all this. So you're pretty much covering your whole brain. Not whole brain, I'm sorry, the whole region where you're inserting your electrodes. However, if you're measuring spikes, you can only measure the spikes of the neurons which are around your electrodes, like here. So there's a lot of things going on here that you're simply not measuring. Your electrodes cannot capture. So what you're looking at is an undersampled, a subsampled version of the actual system, right? Could that be the explanation? So what did we do? Okay, let me make a model. The model is the black dots, okay? This is my cortex, my in silico, my cortex in the computer. Now, can you see a 4x4 electrode matrix here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 electrodes, 4x4. And the red triangles are just the neurons that I can see with those 16 electrodes. The number of neurons in each electrode mimics exactly what we see in experimental data, okay? The exact number of neurons. Then what do? This model is mine. I control it as I wish. I make it critical, okay? I can make whatever I want. It's my model, right? So I make it critical. Proof. I measure avalanches here, and I see a power law distribution. This is the black dots here. The exponent is not 1.5 because it's a 2D network. Therefore, universality classicity changes and goes to 2D directed percolation. So this is the correct exponent, not 1.5. Now, so this is just to convince you guys and the referees that the system is critical. Now, suppose I don't have access to all neurons, like in the experimental data. I sample this critical system because it is critical, because I made it critical, in the red triangles. And I tried to estimate avalanche side distributions from that. What do I get? I get the red curve here, which agrees with experimental data. Right? So, and why this is so? Because, of course, I'm losing the large avalanches because I'm only allowed to see the red triangle. So whenever an avalanche is propagating from here, if it goes into this region here and then here, I'm missing all of this. Whenever if it continues to enrich here, I'm going to see two small avalanches here, not a large one, because I'm missing the whole thing because of subsampling. So this is like, okay, it's not borderline cheating, but it's like really forcing having faith in a theory that you want it to succeed, and try to salvage the theory, right? To save the theory. So this is a way of sustaining criticality, but without proving it. Okay, so okay, with the model you can say that it's compatible, but give me some signature of criticality in your data. A real one. Yes.
Increase the size of the electrodes and redo the process?
You mean decrease the
Decrease the size of
The number of electrodes.
No, just the distance between them, and redo everything you did, right?
Yes, it can be done. Each experiment you have to do, you ask Thais again, and she's there. It's many hours of experiment. Thais, raise your hand, please. I'm talking about you all the time. Yes, she can do experiment. Yes, it's easier to take a large matrix in the first place and then throw away electrodes if you want later. But again, it's my paper. I have to defend it. But this is not. It's not convincing even for us. So I had to look for something else. So since size is limited and our spatial resolution is limited, everything is limited by the number of electrodes, let's try something else. Let's try get some signals of criticality in the time domain. So there are different ways of doing this. The way we first did is a very common way. I'm going to talk about this. Is just look at the Fourier spectrum and the trend fluctuation analysis, which I'm going to explain in the next slide. So we look at the time series of avalanche sizes. So we're looking at some kind of collective phenomena, not one spike, not one neuron, some kind of collective activity like the size of an avalanche. And we're going to, for instance, take the Fourier spectrum or do the trend fluctuation analysis, DFA. For those who don't know DFA, it's a very, very simple thing. It was proposed many years ago. The idea is if you have some time series here, and you bin with some window, this data, and then for each window, you fit the best a linear function, okay? Then you remove your data, the real data from the fitted data, and you calculate the residue of this thing. Okay, this is the residue of your fit. And then you see how this residue increases as you increase the size of the window. You can imagine that the shorter your window is, the best the linear fit will be, because your data has not much time to change much, right? If your window is larger, well, your data can do a lot of things. You're going to fit a straight line. The residues are going to be larger, right? Now, it can be shown, this phrase we love, because we don't have to show, it can be shown that if these residues, as a function of the window size, increases as a power law of the window size with some exponent alpha, then this alpha can be associated to the exponent gamma, which controls the way that autocorrelation decreases with time. Remember, there showed before in the 2 slide courses on phase transition that a normal, typical system, autocorrelation decays exponentially with time, except if it's critical, when you expect the correlation time to diverge and then have something else. For instance, power law decay. So if decay is a power law, autocorrelation function with some exponent gamma, this gamma is related to this alpha, which you can measure here. It's much simpler to measure this alpha than this gamma, like this way. And through the usual theorems that we know all the time, this exponent is related to the power spectrum. So beta is related to gamma through this relation. So essentially, if you measure alpha this way, and you find alpha equals 0.5, this is like a Brownian motion, white noise. There's no correlation in these fluctuations. This is this uninteresting fluctuations. If alpha is larger than 0.5, then you have persistent long-range power law correlations, which again makes you compatible with the critical dynamics. In particular, if alpha is 1, notice what happens if alpha is 1. Put alpha equals 1 here, and you get gamma equals 0, which is very, 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 very long-range time correlation, because it doesn't, even the exponent of the power law decay goes to 0. This is what's called 1/f noise. Okay? So I'm going to use this technique, which is very well established, and look at my time series of avalanche sizes. What do I see? This is the power spectrum. You can look whatever you prefer. It's the same information. Essentially, if you look here, the root mean square deviation, those residues that I mentioned, as a function of the time window, they do scale as power laws, and with exponents which are very close to 1/f noise. This is rat number 4, primary visual cortex, pre-exposure, during waking, during slow wave sleep, during REM. It changes a little bit, but not too much. You get consistent, very non-trivial DFA exponents. This is a different rat, different, slightly different exponents, but not too much. Correspondingly, as the theorems predict, you have power spectra, which decay as 1 over F noise here, exponents 1. Interestingly, if you go to anesthesia, ketamine xylazine anesthesia, ketamine xylazine anesthesia, you don't get anything interesting either here nor here, which is, this is not even a power law to start with. So you miss the temporal signature of a critical system in anesthesia. Keep that in the back of your mind for a while. Then, what we did next is the following. We can look at this time series of avalanche sizes and look at the waiting time distribution. I'm going to do the following. How long do I have to wait before the next avalanche comes? To which I reply with another question. Well, it depends on which size are we talking about. So this is what this SC here is. This is a parameter, which is the minimum size I want to wait for. So how long does it take before the next avalanche comes? Well, it depends. Size 1? Well, I don't have to wait like 1 millisecond. And then another one comes. And then another one comes. If I want an avalanche of size 20, well, I have to wait longer, right? Because they're small. Then another one with size larger than 20 comes. Okay, another one. So waiting times are going to be longer. So if I want an avalanche of size 100, I have to wait even longer. For each value of this minimum size, I have a different lifetime distribution. So I have a family of lifetime distributions. So this is the density of, probability density of having to wait a time tau between the next avalanche of size at least as C comes. This is the quantity defined here. And this is what it looks like. Please look only at the leftmost plot for the moment. The different colors here mean different thresholds. So if threshold is 1, the density of waiting times is biased to the left. As I increase the minimum size, this family of distribution goes toward the right, like I was saying before. It makes sense that if you want larger avalanches, you have to wait longer. And you have a distribution for each, right? And the magic happens when you take each of these curves here, take the red one. The average value of the waiting time is somewhere here. And you can now renormalize the horizontal axis by the average value of here. If you do this for all those curves, which is renormalize the horizontal axis by the mean value, and renormalize the vertical axis by the mean value as well, they all collapse into a single function. What is written here as the dt of SC is 1 over tau, average tau, times some function of this rescale time. And what this means is that information about different values of SC is contained in the same function as a function of only one argument. This is called scaling statistical physics. And it is another signature of criticality. And why is this interesting? Because if I'm looking at time SC equals 1, I'm talking about phenomena which involves one neuron. If I'm talking about SC equals 36, I'm talking about phenomena which involve 36 neurons. The only way they can be explained by a single function is if you are in a critical point with scaling variant regime. That's the rationale behind scaling theory in critical phenomena in the first place, right? So they fall into the same curve. You can repeat this. See, this is animal number 5, awake, hippocampus during exposure, collapse of scaling function. Animal number 4, slow sleep V1 pre-exposure. And animal number 1, REM sleep S1 post-exposure. They all collapse into a single function. They all collapse into a single function. If they are anesthetized, this collapse doesn't work. Maybe another one works, but this kind of collapse doesn't work. So anesthesia is different in the temporal domain. Ketamine, xylazine anesthesia is different in the temporal domain. Moreover, if you do this kind of renormalize the time axis, then it becomes dimensionless. The vertical axis also becomes dimensionless because this density here, I have to integrate on dt to give 1, right? So this has dimension of 1 over t. If I multiply by t, it becomes dimensionless as well. So both axes here are dimensionless. So presumably I can compare different rats now because it's dimensionless quantities. Why not? If you take all those data from different, this is, okay, now different data from one region in waking V1 pre-exposure. They all fall, different rats, onto the same scaling curve, scaling function. Of course, there's some thickness, I know. Please keep in mind these are live animals, okay, not magnetic systems under helium. And again, it doesn't work for anesthesia. And I can even do, and this is a power law, a double power law it's called. This is called a double power law with one exponent here, one exponent here. We can even do this for the different scenario, the 27 possibilities, and they more or less fall into the same scaling curve again, whereas anesthesia doesn't. So this is stronger than that undersampling thing that I told you before, because now scaling is also a very strong signature of criticality. And moreover, this kind of double power law phenomenon is what is also observed in the waiting time distribution. In the time you have to wait for a new avalanche to come, is what is observed in earthquakes as well, with a very kind of shape here. Going back to that very quick notion of universality, we can now try to test very roughly these ideas, because in here I have two exponents, alpha and beta, for each rat, for each situation, for each scenario, and I can try to compare them. In biology, it's always very important to compare whatever you are observing with some random version of what you are observing. There's a control group. So we shuffle the data, etc., and redo everything, and try to force this kind of distribution and obtain exponents. And then when you go to this different scenario, again, 27 scenarios here. Here are just 9 scenarios, right? And look at the exponents you obtain in the different cases and put them in the exponents plane, you see that the raw data, they kind of cluster in the same place, and the shuffle data is spread all over the place. So this is the best thing that I can show you that suggests some kind of universality in the behaviors of the brain in the different kind of regimes, in slow wave sleep, in waking, and REM. Universality in the sense that the exponents fit in the scaling function of the waiting time distributions. They cluster together in exponent space. It's a very indirect way of addressing criticality, but it's one that's very robust in the data, so that's why we're interested in it. Size of the avalanche is a function of what?
Of the rank. So which is the largest avalanche, the second largest one, and so on.
We didn't do this. We never looked at this plot.
But the earthquakes and so on, typically they do that.
You can do that. You can do. Yeah, there are many different. There are many different tricks you can play. Yeah, yes, yeah. This is just one of them. Yeah. So, this is the good news part. So I'm showing you the universality plus long-range time correlations in experiments. So these are two things which are consistent with the critical hypothesis. which is good. I'm talking about experiments now. This is an experimental paper. But as a physicist, I would like to explain this thing. I like to propose a model that can do this. And this is very hard. And remember, this kind of power law distribution that we obtained in the branching process model, remember? You remember what the phase transition was? Absorbing state and non-absorbing state, right? Active versus death versus absorbing. Now we're in trouble here because this kind of model, I create an avalanche and then it goes to silence. And this is what's called separation of time scales, right? What do I mean by hand? You create an avalanche in the model. You create an avalanche and wait until it's dead. Then you start over again and wait until it's dead again. And then you start over and over and over. How on earth, if separation of time scale has to be imposed by hand in a model, by construction, it does not have long-range time correlations because it goes to death, and then you have to start over again. How do you decide when you're going to start over again? You decide, or you put some noise. If you put some noise, there's no long-range time correlation because it's just uncorrelated noise. I'm going to put the correlations in the noise, then you're cheating. You see, we have a problem here. I don't know how to model this. The problem is that I don't know how to model those long-range time correlations. Because the kind of model that I was attached to is kind of makes my life really hard. Because it goes to the non-absorbing state. How can I circumvent the non-absorbing state and go back to... So we have a problem here. Okay? Is that clear? We have a problem? Okay. Waiting time distributions have the same problem. So I'm just exposing you to the same conundrum that we were exposed to. Okay? This paper from 2010. It took us a long time to know what to do. A long time. What time do we have to stop? 20 more? Okay. Let's continue the massacre. Okay. So, pros and cons of that kind of model. This was the first lecture. The things that work beautifully, nice talk, everybody can understand, and lots of hand waving, blah, blah, blah. Okay. Second one, okay. Now, this kind of model, your separation time scales has to be imposed by hand. There's no time correlation between consecutive avalanches, and therefore it cannot explain this kind of data. The most precious data for me, at least, not only because it's my paper, because it's live animals, freely behaving animals. If criticality is supposed to be used for something, it's for live animals, not for a Petri dish, right? So, and I'm seeing interesting things. Compatible with criticality, all right. Complicated stuff, I admit, I'm sorry, but that's what we got. So scaling function, the waiting time distribution has some critical exponents, some characteristic exponents, which are more or less uniform. Okay, it is a signature, whether we like it or not, and I don't know how to explain it, but that's the point. Now, I talked about undersampling already, but I want to revisit you because it's another problem to us. This is what freely behaving data looks like. I've shown you already this data. And this is the nice trick we pulled to try to save the criticality hypothesis, right? So we don't get this, we get the blue line experimentally, and we can get in the model the red line if we undersample like this. So this is a nice trick. We liked it, okay? It's nice. We liked it, the referees accepted, the paper was published, cool, that's nice. I didn't show you yet, but you didn't ask either, about the data from ketamine xylazine anesthesia rats, animals. Nobody asked about the size distributions, avalanche size distributions. You should have asked, but I'm going to show you now. Let us see the size distribution of ketamine xylazine anesthetized animals. Oh, before I do that, just to mention, give due credit to Viola Prisma in Göttingen, who has been saying for a while that undersampling is an important issue. And the reason why undersampling changes avalanche statistics is the way I showed you before. Suppose you have electrodes here and the white electrodes, and an avalanche starts here, okay? So an avalanche starts here, and in next time step, it reaches your neural you're measuring, the white one. So if it's here, so far you're not measuring. So in the full sampling, you have size 1. In the undersampling, you have size 0 because it hasn't yet reached the white one. The next step, it will. Then you're here. Then you measure 1, and here you measure 3, 1, 2, 3. But then it propagates the other way around, the other way, and it goes away from your electrodes. Therefore, your avalanche of size 1 in the undersampling is finished. Whereas the true avalanche has size 3 and keeps going. And then it keeps going. And then it reaches your electrodes here. Then you measure an avalanche of size 2, which is finished, and then it moves on. You see, as the avalanche moves on, and on, you would have, okay, at the end of the day, a true avalanche was size 12. But what you actually measured in your undersampled system was one of size 1, one of size 2, and another one of size 1. That's why your distribution gets all bent down, because you're missing the large avalanches. By the way, you're measuring, you're undersampling. You're not looking very carefully. It's unavoidable. So undersampling solves one problem. Yes?
Data planes in the first experiment, did it have a undersampling in the avalanche?
Yes, good question. So, but didn't Dietmar planes have undersampling in the first? I showed you different kinds of matrices. Remember, in the first paper, it was okay, but those were local field potentials. So local field potentials, you measure a whole thing here. So it doesn't suffer from undersampling as severely as when you were measuring spikes. So we physicists like to confront biologists in the first dialogues when you're starting the collaboration that we don't care about details. Biologists care about details. We're physicists. We don't care about details. Yeah, they are right and we are wrong sometimes. In biology, more often than not, they are right and we are wrong because we do know biology. Because sometimes details matter. And it's a humbling experience for a physicist to recognize that and understand why it matters and learn from that. Okay? So it's a side note. So that undersampling trick with the model was a nice trick. Solved one problem. But it creates another, which is the following. If you look at anesthetized rats with ketamine, xylazine, with the same undersampling, the same electrode matrices, right? So the same undersampling. I do see power law distributions, beautiful ones. Now what do I do with my theory? What do I do with my theory? It was so nice, wasn't it? It was not even a theory. I mean, it was just a simulation, but with some rationale behind it. But now, these are nice power laws. So maybe, okay, maybe if we explore a lot, if we look right in the right parameters, maybe we can get these power laws even when there's sampling. We just tried for that figure in that paper, but let's do another paper just on it, just on this thing, okay? So that's what we did. Okay, let's take Thiago Ribeiro and codes for different kinds of undersampling. Let me show what we did. So the question is, if the model is undersampled like the data is, can it still yield power law side distributions? Because, okay, I didn't want power law distributions here in the free behaving data, but I do in the anesthesia data, ketamine, xylazine anesthesia. See the kind of trouble you get into? So we studied 3 kinds of networks, 2-dimensional networks in which one neuron is connected to a local neighborhood with some local disorder, and then the same thing with some long-range connections, which is what is known as small world network. For those of you who are complex networks aficionados, this is well known. And in the limit of this, some probability of rewiring goes to 1, this becomes a random graph. So 3 different topologies just to cover some basic stuff that could somehow, more or less, roughly mimic the cortex. None of this is the cortex exactly, but okay, It could be roughly two-dimensional with local disorder. It could be random graph. It could be some small world. Okay, let's try the three of them. So for each of these three topologies, I put those simple model neurons, direct the percolation versatility class. I will play with the connectivity until I get the critical point. I will go supercritical, critical, subcritical. I will do avalanches, undersampling in all possible ways that we could think of. And I'll try to find power loss. Is that clear? Methodologically, it's a very simple paper. It's a little bit boring, especially in light of the results. But I want to go through it just because it was a lot of work for Tiago to do. So those three topologies, first, let's be sure that we can control criticality. So again, this is response as a function of stimulus. This is one way of detecting criticality that I showed in the previous lecture. So subcritical regime, you have some linear response. Critical regime, you have some nonlinear response with this exponent 1 over delta H, which for 2D is not 1/2, it's 0.285. You get 1/2 in the small world and the random graph. You can do this way, or you can do the other way, which is just avalanche size distributions. This is the full sampling, okay? I'm sampling the whole model. No undersampling yet. This is just to be sure we know what we're doing. So if you are subcritical with some probability less than PC, you have typically in a log-log distribution, exponential distribution of avalanche sizes. If you are above critical probability, supercritical, you have some bump here with very large avalanches. This is a typical signature of a supercritical system. Large bumps here. This is subcritical, this is supercritical. In the critical case, you have a power law, and a power law.
This is what Strogat's like.
What Strogat's like. This is what Strogat's like. So essentially, I take this 2D network in which you have within some local...
Plus,
yes, it's a disordered 2D plus rewiring, because we allow for some disorder locally, just to be a little bit more realistic. Note that this disorder does not change the right exponents for 2D, so it's robust with respect to that. Okay, so this is just to convince you that we know what we're doing more or less. So we know how to whether the model is critical, supercritical, subcritical. Now we're going to undersample the three of them in all three regimes and hope for the best. Yes?
Just a quick question. On the distribution plot,
Yes.
right-hand side, small green dot that appears outside of the power law in every single, up. Yeah. So every time this point is always there in D, E, and F, right? So every different case.
Yes.
So which means it's a systematic thing. It's not just one green.
Yes.
It's outside of the power law, right?
Yes, yes. It's a supercritical case, because this is the P larger than PC, so it's a supercritical case. The supercritical case means, remember from the animation, it's when you have this complete the dominance of the self-sustained state. The non-zero activity becomes stable. It doesn't always happen. So if you take a supercritical network and you just create one spike, it could be that it will die out. It could be. Try it again. It could be that it will die out. So this is what gives you this distribution here. This is when activity somehow stopped. But it will also happen in a finite probability that it will stay there forever. So this forever, it corresponds to a huge bump here of very long avalanches, because you have to stop your simulation at some point. That's what those bumps mean. These are not a critical regime, they're the supercritical regime.
It looked just way off.
It is off because it is a bump. It is a bump which corresponds to the self-sustained activity. So if you measure self-sustained activity, there's no silence. So this one huge avalanche, which is why the bump is so far away in log scale. You're right. Okay? So now we undersample this mess to get more mess. So you can vary, for instance, you can keep your network here and keep the spacing, say dm equals weight, which means 8, the distance between the electrodes is 8 sites. which is more or less what corresponds to the reality in biology. When you look at the spacing between electrons, 150 microns with 10 to 15 to 20 microns radius of each neuron, that's what more or less what you get, say 8 cellular bodies. It can fix this distance and change the number of electrodes, for instance, okay? And then I promise that this mess is going to turn out very simple for you, because here I'm showing the 2D, this column is the 2D graph, 2D. This is the small world topology, and this is random graph topology, okay? This is supercritical regime, this is critical regime, and this is supercritical regime. And the different colors are different number of electrodes. And the question is very simple. I know it's very complicated. There are many situations here. All I ask is please tell me whether you see any straight lines in those plots. And the answer is that you should see 3 straight lines. Can you confirm that, please? That you can see only 3 straight lines in those distributions. Do you all agree? Yeah? Can you all see 3 straight lines? This one, and this one? More or less here. Yeah? The rest are not straight lines. Therefore, they're not power laws. And which are these 3 lines? Black dots, full sample. Yeah, but there's no undersampling here. For the full sampling, for the full sampling, I do get power laws. For any way do this undersampling, I don't get power laws. Well, maybe it's because I'm just changing the number of electrodes, but I'm not playing with the density, with the distance. So let's change that. So instead of changing the number, I keep the number and I change the distance. Right? Okay? Again, 2-dimensional topology, small world topology, random graph topology, supercritical, critical, supercritical. Please find me the 3 straight lines. They are the same ones. Which is precisely an interesting case in which there's no undersampling. I don't find power laws, even if the system is critical. Here's this, everybody's critical here. I don't see power laws. So, like I promised, this is a hard lecture, the second one, because it's about things that are going wrong. So, separation of time scale has to be imposed by hand in the model. Therefore, no time correlation between consecutive avalanches can be reasonably expected as they are observed in the experimental data. So I don't know how to reconcile that kind of model with the experimental data we get in the lab. Moreover, when I try to explain some of the data, this banding of the size distributions with undersampling, which I see in the freely behaving data, that's very nice. The trick works. Undersampling works in my favor. Now, when I realize that with the same undersampling, the data of ketamine, xylazine, anesthesia gives me power law distributions, then undersampling works against me because now I cannot explain it. That model cannot explain it. Then we're in trouble. So you see that the message here is kind of mixed. You have some signatures in the free behaving animals. Anesthesia complicates matters. There's something about undersampling. Where does that leave us from this point, from this perspective of this criticality thing? The answer is that leaves us perplexed, at least me, us, our group, our colleagues. We didn't quite know what to do with this thing. So we had no model for these long-range time correlations. And we didn't know how to proceed. Why? Why? It's interesting to discuss why. Because in the first result, we get this 3/2 exponent. It links us immediately to the branching process. It's very easy to explain. It's a very appealing model. So sometimes we get attached to models because they're so good, they're so beautiful. And it's very easy to get attached to models. Even when the data is screaming in front of you, that, hey, there's something wrong with this model. But the model is so beautiful. It's a mistake that it's very easy to make We made it ourselves. And how to get out of this conundrum? Tomorrow, right? Tomorrow. Okay, thank you. Of course, questions anytime. Yeah.
Did you expect power laws in the anesthesia ones?
What?
Sorry. Did you expect power laws in the rats with anesthesia, or it just came up?
No, we did expect, because I went through very fast, but in my first lecture, when I first talked about neuronal avalanche, this is the first lecture, bags and plants. So they first got in a slice, and then the first thing that they thought of, okay, let's go to an intact animal, a whole animal, in vivo, not in vitro. And they did, blah, 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 hold on. There you go, here. They had already, I don't remember which anesthesia is this, but they already had power law distribution in anesthetized rats with local field potentials, which is this electric potential that covers a large region here. We expected power law distributions in the spikes for the free behaving animal in the first place, and they didn't show up. So then we tried, okay, let's try anesthesia. And then, okay, at least it looks compatible with this, at least. But we're in trouble with undersampling. Ketamine xylazine anesthesia.
Yeah,
Ketamine,
no, I mean, you use ketamine with psilocin when you have to do an intervention where you don't care about the perception of the animal. That means, for example, if you want to do just a deprivation of the pain, but you don't care whether the animal would be perceiving something from the sensory input, then you do ketamine with psilocin. But if, for example, you want to expose the bird to a song, for example, and you want the brain to capture this information, you use urethane,
yes,
which is anesthesia the bird will never recover from. But then you are exposing the brain to some input from the exterior. So
yes,
when you use psilocin and ketamine, then you are closer to a slice preparation. And when you use urethane, you're exposing the brain to a set of stimuli that will set it into some of these lower dimensional, patterns of activity. So
yes,
there is a key relationship between the kind of anesthesia that you have and the kind of expectation that you might have. I would have also thought that with ketamine and psilocin, you are more likely to have something that dynamically is closer to a preparation than with urethane.
And this is just a comment, right?
That's a comment.
Yes, and thank you for this comment, because please note what he said. So ketamine xylazine is this kind of anesthesia which deprives the organism from sensory inputs, whereas urethane is different. It's a different kind of anesthetic, which in his case in the lab with the birds allow the birds to receive income stimuli. Why I mention that? Because in my fourth lecture, we'll go back to urethane. Urethane will be my hero.
Your friend.
Yeah, my friend. Yes, yes. I'll make peace to many of those questions here. I promise. It's a promise to you guys. Yes. Keep tuned. No spoilers.
Some other question?
Yeah.
Hi again. So I'm not a biologist, right? I'm a mathematician, and I'm looking at these things, and I found them really interesting.
You're a mathematician?
Yes.
Okay.
So all of these things are kind of new to me, apart from the probability distributions that you're showing.
Okay.
My question is, like, if you are expecting power laws in anesthetized rats, but you find different distributions for, let's say, freely behaved animals, could it be that the animals are described by power laws when they are biased in some way? So, for example, when they take some substances that you describe, or some drugs or something like that, could it be that every, let's say, because now you're talking about different types of drugs, which I'm not familiar with, but they are described by these kind of distributions, right? Power laws. But then when they are freely behaving, you find these log normal things.
Yes. Yeah, that's a very interesting question. It goes back a little bit in a very twisted way to what I jokingly referred to this Heisenberg principle of biology. The Heisenberg principle, it's like in the quantum mechanics that if you squeeze momentum, you lose position, vice versa. It's a little bit like this. Okay, ideally the animal should be free because then you can observe it as it is. But then as it is free, then you cannot control it. And sometimes you need control in order to measure things properly. To measure things properly, then you anesthetize the animal, and you can measure for long hours. But then the animal is anesthetized. What you're seeing is not a true animal. So it's a common issue. It's a trade-off that has to be done in biology. So the way you ask your question, it's very bold in the sense that it calls into question the whole theory.
It does? Sorry about that.
No, there's nothing to be sorry about. It's an important question. Because if I can only see signs of criticality in slices or anesthetized animals, then what's the point? It challenged the whole point that I started with in the first place. That criticality is nice, blah, 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 because it's complex, it can handle external stimuli, blah, 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 right? If it only works in the slice, well, slices don't do much, don't have to survive or face the world.
It could be that it's an adequate description for some states, and then for other states,
yes.
You change your parameters and your dynamics and go into a different state. So why would they expect it to be true for every moment of the
Precisely. So there's that. There's this. Like I said, please keep in mind the golf course and the hike in the woods. Theoretically speaking, right? It's very rudimentary ways of trying to approach theoretically a problem which is very complex. We don't have a Hamiltonian from the brain. We don't even know which are the minimum ingredients. We don't know where to coarse grain our systems. We're trying. Everybody's trying. If you go to the Society of Neuroscience annual meetings, like 30,000 people, we're trying to do this. Could it be that just fMRI is enough, just 1 cubic millimeter resolution, but then you lose the time resolution? I'm not giving you a direct answer. I'm just saying that's a very good question, and which goes directly into the crux of the question here. We don't know. It's just a conjecture, and with many of us think it's interesting enough to try to probe it. We try to probe it here, probe it there. Then it fails. It fails with the free behaving animals. Well, but then we fix it with undersampling. Well, then undersampling caused me trouble in anesthesia because I don't know how to model. So we try to run after this rat, this animal, and it could be everything that you said. It could be the.
In my mind, I see, let's say, the state that the rat is feeling well as, I don't know, the value of a parameter, and then the state that where it's biased with the value of this parameter being different. Then I can have, let's say, a family of distributions based on this parameter. And then for each value of this parameter, I can have a different distribution describing the state at which, let's say, the rat will be. And there is a theory called superstatistics that does this kind of thing. If, let's say, you have this phase space of the possible state at which you can find the rat, and each one of these points is described by one distribution, you can probably describe the case where the animal is biased or not biased or some other state which I'm not aware of.
Okay.
Just my thoughts, that's all.
That's interesting. If I understood correctly, it might relate indirectly to what I'm going to talk about in the fourth lecture, where we're going to revisit with urethane, my friend anesthesia. No, I don't use it. No, you don't use it. Never, you cannot come back from it, okay? It's one time only. And where we essentially stop talking about the animal as just being an animal, like in anesthesia state. Even if you're in anesthesia state, or especially you retain, you can go through different states, which I'm going to define properly. And then if you look across, like you gained one axis to explore. Then matters become more clear. It's almost a spoiler. I should shut up. Yeah. It's a good question. Thank you.
Other questions?
No.
So
Great,
we
thank
finish our lecture today. So after a break, we have the project assignment. So you have to group in 4 people. Humberto Shandira will send you, or have sent you, I don't know, a Google form just to fill the group and the title and the paper you choose, so in order to have a record of this. So I don't know